Hello again. We are back with chapter 14. This is going to be about how biological diversity evolves. So we're going to be looking at a larger scale than the microevolution from the last chapter. Now, a lot of these are sort of, classic. You see the picture of Darwin, the Neanderthal, um, and say, well, what about the kid with the corn? Well, we've bred, all sorts of organisms for our own purposes, and often they don't look very close to what the originals did. Now, again, when I talked in the last chapter about some of the evidences of evolution, things like the fossil record, right? So you can find the history of living things by investigating layers of what are now rock where they used to live. Now, we've had a huge effect on the geology and ecology of this planet. One interesting side effect, if you will, of the COVID pandemic was seeing the change in atmosphere over places that were typically very polluted. So it gave everyone a very strong impression of how much we affect it and how rapidly it can bounce back if we change our ways. Anyway, things that we do that last. We move organisms far from where they came from, right? Either as pets or for food or for entertainment. We have changed a great number of plants and changed the ecology of many locations because of our use of agriculture and again with domesticated animals. We've bred these animals selectively into forms that are often quite different from what their natural ancestors were. And of course, we manufacture things, right? We create things like plastic and concrete, some of which will take a very long time to ever break down back to the original state if we don't find a way of degrading it, particularly plastic. Lasting radioactivity from nuclear weapons and, as you now know, accidents at nuclear plants like what happened in Japan. And of course, the climate altering emissions from burning fossil fuels. Now, the fact that some of these changes are irreversible. Now we have hope that we can try and ameliorate the climate stuff, but as far as plastics and some of the other things we've done, as far as driving some species to extinction, changing some environments that in ways that will never likely recover anytime soon. Now they've decided to call this a new epoch. Now, if you go back, you've got things like you hear of Jurassic Park, right? So that was part of what's called the Mesozoic era. Right? There's names for all these periods of time. Well, they're actually proposing a new one, the Anthropocene, that our effects on the planet to the level of the geologic record. So people who would be investigating many years from now would see suddenly a drop off in species, basically an extinction event in many respects, and all the other things we've done. That it will be a distinguishable period of time in the history of Earth of the effects that we've had upon it. And of course, there's, our actions within the environment also bring about evolutionary change. Right? We're talking about the pesticide-resistant insects and antibiotic-resistant bacteria before. So we definitely changed the world. Now, in the 150 years since the publication of Darwin's On the Origin of Species, new discoveries and technological advances have given us a lot of new information about the evolution of life. Remember, back then, there weren't, no one knew of DNA, no one knew of microanatomy. There was so much that wasn't known back then. So the premises that Darwin laid down were great, but it's interesting that we can still apply these ideas to living things, understanding living things, including with all the new technology we have. Now, how did we get the diversity of life? How did we get from the very simple single cells or very simple colonial organisms that were in the ancient seas? Well, we call this process speciation. And this is the process by which one species splits into two or more species, right? But I'm always going on about the iguanas, right? So on the left side, you see the classic terrestrial iguana. On the right, you see a marine iguana. Now, from anatomy and now more recently genetic study, we know that those marine iguanas evolved from the regular land iguanas, regular green iguanas that had basically been blown out. Sometimes there's something called rafting, where an animal is on a piece of wood and gets, drifts out to a new location because things like reptiles don't need as much food energy, so a lot of them can last through unintended voyages like that. So if you look at this iguana closely, you can see, okay, he's got these long claws because he climbs trees, and he's got coloration to sort of blend in the tree. And this is a series of features that most of you guys have seen iguanas. Now, when you look closely at the Galapagos marine iguana, you can see, oh, yeah, I can still see the frill on the back, and I can still see the long nails, but the head's different because now this is something that's not grabbing leaves off of branches, it's scraping algae off of rocks. And the tail is basically turned into a paddle. It's very flattened, vertically, whereas it would look almost triangular in cross-section if you looked at a regular terrestrial iguana. So there were things that happened that changed the descendants of those iguanas that got blown out to the Galapagos from the iguanas that, whose descendants remain on the mainland. Now, the term species, it's a Latin word that literally means kind or appearance. The biological species concept defines a species as a group of populations whose members have the potential to interbreed with one another in nature and can produce fertile offspring, right? So offspring that can themselves reproduce. So if you think of some of the things we do in captivity, like crossbreeding, donkeys and horses, or probably a better example would be lions and tigers, to create hybrids of those animals, those would not, the offspring would not be a biological species because they don't interbreed with each other in the natural world. They often don't even live in the same area in the natural world. So it's the sort of thing where if we do narrow down when a species is by this functional element, can they breed naturally and would they be producing fertile offspring? So now you can't apply it to things like asexual organisms, right? If there is no interbreeding when you're reproducing on your own, you can't apply it to fossils because, not being alive, fossils don't breed, right? So we can apply the biological species concept to living things and things that reproduce sexually, but obviously not in all situations. And it's an interesting thing because it's based on reproductive compatibility rather than physical similarity. If you look on the right-hand side, you see a picture of a variety of people, right? We have quite a lot of morphological diversity within our own species, but of course are all still reproductively compatible. And you look on the left-hand side and you see a couple of birds, and those are actually two different species, even though they look very similar at first glance. So the biological species concept, again, comes down to reproductive compatibility rather than physical similarity. Now, again, how do we define a species? Well, biologists may also define a species not just by the reproductive elements, but according to measurable physical traits. So things like bone or skin, or looking at how the digestive tract is put together. There are things that you can compare and link things together by their anatomy. You can also take it all the way down to molecular data, right? Because when your book says it's kind of like a barcode that identifies each species, so you can have two species that look very similar, like these birds back here, but they are actually different species. And so maybe at one time in history, they might have been linked together as the same species, but once people started mapping out the genetics of various species, they realized, no, these guys are not that closely related. Also, a species can be described as the smallest group of individuals sharing a common ancestor and forming one branch on the tree of life. So moving on to what is it that keeps things from interbreeding, right? So we keep saying, okay, things that can interbreed our species. Well, why is it that things don't interbreed with each other? We call these hurdles reproductive barriers. So reproductive barrier is anything that's going to prevent individuals of closely related species from interbreeding. Now, if things are very distantly related, there's going to be all sorts of incompatibilities, but things that have not diverged a long time ago, things that have not separated in species that long ago, they still may have a lot of similarities. So what is it that stops it? Well, it breaks it into two major categories: prezygotic barriers and postzygotic barriers. And as you can guess, pre refers to before, post refers to after. So this is things that would occur before a fertilized egg is produced or after a fertilized egg is produced, right? The zygote is that first cell of a fertilized egg. So prezygotic barriers prevent mating or actual fertilization between species. The postzygotic barriers operate if interspecies mating does occur and hybrid zygotes are formed. So things like behavioral isolation, which we'll get into in a moment, okay, this would be a prezygotic barrier. If the two organisms don't interact with each other for one reason or another, well, then definitely you're not going to have any mating or zygotes forming. And so you have this big, long diagram here. And so again, these are reproductive barriers between closely related species. And as you can see, the top part have the little no symbols in blue. Those are all prezygotic. Below, roughly the midline, you see all the postzygotic barriers in magenta, right? And then if there are no barriers have stopped interbreeding, then what do you have? Viable fertile offspring, right? So that would be successful breeding between closely related species. So we're going to walk through these guys. I'll go through them briefly here, and we're going to go through them again in some of the later slides. So again, those two dots at the top represent individuals of two different species. So prezygotic barriers would be temporal isolation. They might live in the same area, but one is active at day, one is active at night. Right? They won't run into each other, so they would not naturally interbreed. Habitat isolation. You could have two species living in the same sort of area, but say, if you think of a forest, one living high in a tree and one living on the ground. They may be active at the same time in the same place, but they're not in the same location. And finally, behavioral isolation. Say they do live in the same sort of area and they are active at the same point of the day, there's going to be something that keeps them apart. And so things like the dance of the blue-footed booby, which would not do anything for the albatrosses that are also found on the islands where they live. So we'll get to these in more detail shortly. Let's say you overcome all the three barriers, right? So you actually get a mating attempt. The first the next barrier is simply mechanical isolation. The parts don't go together. If you look at a lot of invertebrates, like many spiders, insects, a lot of arthropods, when you look at the complexity of some of their reproductive structures, you can realize that literally parts won't go together. So if mechanically can't mate, well, that would definitely be a prezygotic barrier, right? You wouldn't have the ability for the sperm to make it to the egg. The next would be gametic isolation. So maybe mating did take place, but the gametes can't fuse properly. Well, let's say we got past all of those barriers. So what could happen? What are barriers that could happen past this point? All right, so zygote forms. So now everything after this would be a postzygotic barrier. Well, you can have reduced hybrid viability. In other words, the hybrid that forms, you have a zygote forms, but the hybrid either does not make it to birth or hatching, or it's fairly weak and just doesn't thrive, right? The next would be reduced hybrid fertility. So say, okay, the hybrid is born, grows up, makes it to reproductive age, but sometimes there's issues of sterility where they can't reproduce with another organism. And hybrid breakdown. Sometimes after, you might have enough fertility for reproduction, but the offspring of the hybrid don't do well. If none of these barriers were present, then you have viable fertile offspring. You've got yourself a hybrid between two species. So going into the prezygotic barriers, right? So we're going to talk about each of these briefly. So part one, temporal isolation. We've got here spotted skunk and we have two species of skunks here. and they don't mate at the same time. So I talked about things being active in the day or the night. Sometimes organisms breed at different times of the year. So they may live in the same area and see each other, but they're literally not, you know, they don't go into reproductive condition at the same time. So you can't have cross fertility because they won't both be fertile at the same time. Habitat isolation. So here's a couple of closely related garter snakes, but one lives, as you can see in the upper right, one lives in sort of a dry territory and the other lives mostly in the water. And the coloration is, you can actually see the coloration is sort of matches the environments they're living in. So they may live in a similar place, but they just aren't going to run into each other. And of course, my favorite, behavioral isolation. So many birds have very elaborate songs or dances or creating structures to attract females. There's all sorts of ways in which typically males are attracting females, usually it's that pattern. And so what looks like a lovely dance of one blue-footed booby to another blue-footed booby is sort of, what's that guy doing over there to, say, one of the albatross species on the island. So they might be in the same place, same time, but what one does doesn't do it for the other. Now, there's things like mechanical isolation. This one's kind of interesting. Many plants are pollinated by organisms, right? Birds, insects, even lizards, things like that. The plant on the left has that hummingbird that acts as its pollinator. So it feeds from plant to plant and spreads the pollen that way. That hummingbird can feed from that plant on the left. The bee can fit down into that flower in just the right way so it can get what it wants to eat and pick up the pollen as it does so. But it won't fit on the right flower. The right flower's anatomy is not favorable to that hummingbird being able to get in there. So this is something where the mechanical isolation isn't directly organism to organism, but in this case, the intermediate, the pollinator, is not compatible with both of those two species. And gametic isolation. Think about it. Some organisms reproduce in the water and just dump their gametes, right? They release the eggs and sperm into the water, and they fuse there. Many fish, when you hear fish spawning, right? It's not internal fertilization. So this is where gametic isolation becomes really important, right? If everyone's dumping their gametes in the water, there's a higher chance of gametes from two different species running into each other if you have things that are spawning at the same time of year. So you have these sea urchin species whose gametes cannot fuse. So they may be releasing their gametes at the same time, but the gametes won't fuse, so it keeps the species separate. Now, say you get through all that and you actually have a zygote form, right? So what are the things that can be problematic after that point? And again, reduced hybrid viability, reduced hybrid fertility, and hybrid breakdown. So in this particular instance, it's a type of salamander where you may have interbreeding between these two closely related species, but the offspring is fragile. It's less likely to survive to adulthood and be able to reproduce itself. The other is, okay, they make it to adulthood and they're stable, but they're not fertile. Now, this is a classic example, right? Horses, donkeys and horses can be bred and you get what's called a mule. And classically, every book would say the mule is the sterile hybrid of the horse and the donkey. Well, of course, science loves to, find out that they were not totally correct. There are cited examples of fertile mules. And so somewhere somebody is probably doing the research. It's like, why, it may just be, reduced fertility. It may be just some chance that whatever combination of genes allows a mule to be fertile. But it's just kind of funny, in the last, say, 10 years, all of a sudden they've actually found instances of fertile mules. So never say never. Now, post-zygotic breakdown. So you have a hybrid, it succeeds, it gets to maturity, it's fertile, right? It can reproduce. But that next generation, right? So the hybrid of the two species, when it reproduces, its offspring, right? The F2 generation, if you will, is sterile. So there's points at which things don't go properly. So
The waved albatross is one of the largest birds in the
here's
Galapagos. These waved
the
albatrosses are
waved
performing their
albatross.
courtship ritual.
They do
Notice
sort
the
of beak
behaviors:
fencing and then
sky
sky
pointing with head
pointing
held back, beak
and
clacking,
all sorts of
and heads
noises.
bobbing left and right. The complex courtship dance, with very ritualistic movements, can take a long time to complete. Albatrosses pair for life, up to 40 or 50 years in some cases. Most of the entire world population of the waved albatross nests on Espanola Island and is considered endemic to this Galapagos island. These birds land on Espanola Island in late March and leave before January to wander over an extensive range along the South American coast from Ecuador to Peru. Credit Richard Benz, Wycliffe School.
Now, in comparison, we have also living on Espanola.
The Galapagos blue-footed booby, Sula nebusiae, is the most common booby in the archipelago. It nests near the coast of most of the islands and feeds close to shore. This video shows blue-footed boobies filmed during their courtship ritual. You can see the birds displaying their blue feet to each other and holding their wings back while tilting their bills up, a behavior called sky pointing. Although both male and female are approximately the same size, they can be distinguished by their vocalizations and their eyes. The female's voice is a definite honk, while the male whistles during courtship. The female's eye has a noticeably larger pupil. In this sequence, the male booby is the one seeking the female's attention with the sky-pointing behavior. Credit Richard Benz, Wycliffe School.
Right. So you can imagine if these birds were to encounter each other, the rituals are so different that it would probably have zero chance of succeeding, in terms of a mating. And there's other courtship rituals.
These two
right?
giraffes on the Serengeti Plain
male
in
giraffes
Africa
trying
are
to
bumping each other's necks in a courtship ritual. Courtship usually involves a complex set of behaviors believed to serve as confirmation that the participants are of the same species but of opposite sex, are in good physiological condition, and do not threaten each other. Credit National Geographic.
So that's not so much a combat over a female, it's sort of a making sure that this other individual is an appropriate mate. Okay, so mechanisms of speciation. So a key event in the origin of a species, right? Two or more species diverging from a single species. This happens when a population is somehow cut off from other populations of the parent species. And there's two ways of describing this. One, which is the most common, is allopatric speciation, in which the initial block to gene flow is a geographic barrier, right? Allo meaning different, patric meaning, think of country, right? We talk of patriotism. Well, allopatric is different location. So the initial block is the geographic barrier that physically isolates the populations from each other. Or sympatric speciation, sim meaning with, these are formed without geographic isolation, and they're much rarer, and I'll go into some examples later on. Now, things that can physically isolate populations, geologic processes, right? So you have this initial species, but when they're physically separated, right, they may develop into develop somewhat different characteristics to the point where the reproductive barriers from the preceding list would keep them as separate species. Right? And here's a great example. These are antelope ground squirrels, and they are found on opposite rims of the Grand Canyon. So that is one heck of a physical, barrier between the two. So over the many, many, many years that the Grand Canyon's been forming, these guys have been obviously more and more separated to the point where now, let's say you put up a bridge or something and the two species could cross, they no longer interbreed. Now, sometimes things happen, and sometimes it is something like humans making a bridge, right? So if something were done that connected these two populations again, once they've diverged, there's two possible outcomes. If you look at the upper one, okay, so at the beginning on the left-hand side, you see a single line, and then the point where the line splits, right? That's where the populations become allopatric. And that thing in between is the geographic barrier. Well, let's say something where the populations are separated and they keep extending, and maybe they extend past the point of the physical barrier. So if they started interbreeding with each other and the gene pools merged, right, that they did succeed in breeding with each other, then you'd say there was not true speciation, right? Because they're able to reproduce naturally in the wild, and there's no barrier. If you look at the lower illustration, if these guys diverge and get past that geographic barrier and encounter each other again, they can't interbreed, right? And for, again, one of those reasons, one of those prezygotic or postzygotic barriers, well, then you've got reproductive isolation and speciation has actually taken place. Just gonna pop past that. Okay, so sympatric. This is the weird one, right? Because you're thinking, okay, allopatric, something separates them, right? Iguanas go up to an island, ground squirrels live on opposite sides of a growing canyon. There's something that physically separates them. So in sympatric speciation, a new species arises within the same geographic area as the original parent species. Now, you'd say, okay, well, what stops the gene flow then? How is it they don't keep interbreeding? Well, there's three basic forms things that can take place. One is polyploidy. Remember, we use the term haploid and diploid. Well, polyploid is what? Something that's happening where you wind up with an extra set of chromosomes. So sometimes a species may originate from an accident during cell division that results in a polyploid cell, a cell that has extra sets of chromosomes. Sometimes habitat complexity, the habitat changes, and the organisms sort of subdivide the habitat and become, they're still in the same geographic area, but they start living in somewhat different habitats, like I talked about before, in a forest, something living high in a tree and something living on the ground. So they're still in the same area, but they've diverged in where they live. And sexual selection. Sometimes things change in, we saw the boobies and the albatrosses a few minutes ago. Well, things can happen that change sexual selection in organisms. And once you have that taking place, you can have two populations that, because what causes the females to choose a mate, it's typically females choosing males, changes. Right, and so what looked good with the ancestral species now it doesn't look quite so good, right? So you now have a divergence. So still in the same area, but something changes, and so they won't select, the members of what is now the new diverging species. Now, polyploidy, you might say, well, that doesn't sound like something that could be good, right? You wind up with extra sets of chromosomes. Well, typically we see this in plants. And there's two distinct forms of polyploid speciation that have been studied and observed out there. One, polyploidy arises from a single parent species. All right, so think of a failure of cell division, where you go from a 2N cell to a 4N cell. Right, so you have a big old fail in cell division, so now you've got these unusual forms. A second form of polyploid speciation can occur when two different species interbreed and produce hybrid offspring that can't interbreed with either of the parental populations. So you can have a hybrid that is still living among the other individuals, but can't breed within the individuals. And you might say, well, this still doesn't make sense. Well, remember I said a while back when I was talking about Mendel, some plants can self-fertilize. So if you've got a plant that can reproduce asexually or fertilize itself, then you don't need a second individual. And a lot of the plant species we grow for food are actually polyploidal. Oats, potatoes, bananas, strawberries, peanuts, apples, even sugarcane, and our most commonly used wheat. So we actually have taken advantage of these polyploid organisms. So just to show you an example of how this works. So you've got species A, 2N is 4, the diploid state is 4. Species B, the diploid state is 6. All right, so go to the next illustration. So if you are undergoing meiosis, right, you're having reduction division. So you're going to go from 2N equal 4 cells to N equal 2 cells, right, half the genetic complement. So n equals 2 in the gamete on the top, and n equals 3 for the gamete on the bottom. So you're saying, oh, this is not a compatible number of chromosomes. Well, if you see something here where, okay, the odd number of chromosomes can't pair, you'd think, oh, all right, game over. But again, if it's viable, you now have a sterile hybrid. It can't breed back to either parent, but if it can reproduce asexually, you can have more individuals. Now, if you get a cell division error at this point, now you have the chromosomes doubled, and you wind up with another strange hybrid that can't breed back to either its parent generation or its grandparental generation, if you will, and you've got 2 unequal to 10. So it's the sort of thing where, again, you really don't see this much in animals. This is more of a plant thing because, again, many plants can reproduce asexually. And so in the middle of a field, you could have a polyploid individual develop and reproduce and be there, a new species amidst the species that it arose from as a hybrid. Now, do we facilitate speciation, right? Do we introduce things to organisms to each other that otherwise would never have encountered each other, right? As we run all over the planet, we're often taking things with us, plants, animals, other organisms, to areas where they would never have moved naturally. So now you've got opportunities for hybridization that otherwise would not have happened. And they have an example here. 50 years after the introduction of the plant goat's beard, it's kind of like a daisy, it's in the Asteraceae. After the plant goat's beard was introduced to the United States, a botanist realized that hybridization had produced 2 new species from that original one. And so they conducted tests to determine how one new species formed. Right, and this is Tragopogon murus. Well, the results supported the hypothesis that this developed by polyploid speciation. Now, I'm not going to ask you about the details of this on the test. I just want you to see an example of sympatric speciation, or I'm sorry, speciation from humans moving things around. So you can kind of see, if you look at the illustration there on the left, they thought that a new species of goat's beard formed by polyploid speciation. And the thing is that what they did was they manipulated these organisms to encourage chromosome doubling, right? Because it would take a long time to wait to see this in the wild. So they actually treated the seedlings with a chemical that tended to double the chromosomes to see if they could simulate what happened in nature in the laboratory. And sure enough, they were able to develop polyploid plants. If you look at the little pie chart in the upper part, let me get to a bigger picture of that. All right, here we go. So polyploid plants resulting from hybridizing those 2 species were consistent with the new species that had been discovered and was suspected to be a hybrid. So they were able to simulate the conditions that led to that hybrid being formed in the first place. So in that, the self-pollination, you saw, okay, one form developing there. 20% the seeds were not viable, right? They didn't germinate, right? So that would be some sort of post-zygotic event. And then, a little over 1/3, you wound up with polyploid hybrids. So again, this is something you mostly see in plants because polyploidy usually is a titanic fail in most organisms that reproduce sexually. Okay, so back to the Galapagos. Also, the Hawaiian Islands, right? We always hear of Darwin and the Galapagos, but the Hawaiian Island chain is also another really good place to observe how species operate. Both of these were volcanic island chains. So what happens is as the Earth is shifting, you wind up with lava coming up, and if there's enough lava coming up, you wind up with these islands. Well, over time, it's like if you think of an escalator. If you've ever been to a store and they have an escalator and you have the stairs that sort of go up and then they slide underneath the floor, when you get to the top. That happens to island chains, right? They form by volcanoes, so like the stairs popping up at the bottom. And then when you get to the top, it's kind of what happened is something called subduction, that those islands eventually get tucked back under the crust of the earth. And so the islands are formed and eventually disappear. So it's sort of like an escalator you'd see. So during the time they are there, when they first form, right, you've got something that formed from molten rock. Nothing's going to be living on lava when it first, comes up. So eventually it cools, and then it becomes a place where life can develop. Life can arrive. So ocean currents, like the iguanas that get rafted out on a piece of wood, or winds, could be plant seeds, could be spores, could be birds that get blown away in a storm, right? So one way or the other, colonists arrive at these brand new islands. In that new environment, their populations can often change massively from the distinct parent populations, because there might have, let's say, the parental population lived in a certain environment with certain food plants, etc. Well, those food plants might not have conveniently wound up on that new island. So they have to learn how to deal with new food sources or new habitat to dwell in or breed in, etc. In addition, islands that have physically diverse habitats and are far enough apart to permit populations to evolve in isolation, but close enough to allow occasional dispersals to occur, are often the sites of multiple speciation events, right? So they're close enough where things can still occasionally, spread from one island to another. Most of the time we tend to talk about the Galapagos, of course. So these islands have all these closely related finches that came from common ancestors, right? So there's 14 species. Now they share many standard finch-like behaviors, but they differ in their feeding habits, the habitats where they live, and their beaks. Bird beaks become specialized for the lifestyle of a bird, because the beak is the primary tool by which birds get their food. So the distinctive beaks that you see have adapted to the specific diets of the different species of finches, because they evolved in these different habitats on these different islands. So you might have a common ancestor blown out to a series of islands, but if the plant life is different on those islands, they often will diverge physically to adapt to those environments. Now, the fact that you see these distinctive beaks that link to their diets, again, we're back to structure and function. So you have a couple of examples here. The one at the top it feeds on cactus seeds. Right, so it's got a big, strong bill for prying those seeds out of the fruit. And they call it a cactus finch. That's convenient. If you look to the right, you have an insect eater. Right, if you look at that beak, it's kind of like a pair of tweezers, right, for grabbing little bugs. Right, so it's not like you're trying to mechanically pry a big seed out of a tough fruit. The point of the guy on the right there is to be able to grab little insects efficiently. And if your beak doesn't change enough, there's also something called tools. And we tend to think of tools as a human thing or something we also see in great apes. But there are other animals that have developed tools. And in the lower left, you have the woodpecker finch that eats things like grubs and wood. And it just can't reach to the point of pulling the grubs out of their little tunnels. So it's learned how to take pieces of twigs and fish around in the hole, stab that grub, and pull it back out and eat it. So it's kind of neat that, okay, to a point, beaks will get the job done, and sometimes it's the ability to have behavior that adapts to the environment. So in contrast to the microevolutionary change, that generation to generation change I talked about in the last chapter, this process of speciation, at the larger level, is often really slow, right? So you're not looking at generation to generation. You're often looking at generations to generate it, to show a change. But you can see speciation happen, and it's kind of interesting. Researchers have now documented a lot of cases where populations are currently diverging as they either use different food resources or breed in different habitats. So something that people can observe in in human lifetime scale. Now, usually when we talk about evolution at the macro level, we're talking about the fossil record, right? And somebody did a survey of 84 groups of plants and animals, and the time for one species to diverge into two, to split into two, ranged from between 4,000 to 40 million years, right? So some changes are wicked slow, some are, in geological sense, relatively fast. So the cumulative effect of all the speciations of all these different organisms on the planet, as well as extinctions that took species out, have shaped all the wild changes that you can see going through the fossil record. So the fact that you see a planet that's dominated by giant reptiles, and you have a giant extinction event, and all of a sudden you've got a lot of job openings for all the little mammals, and so the mammals then diverged into many forms. So extinction events are another thing that promotes speciation. So macroevolution is evolutionary change above the species level. So micro is within a species, right? Change generation to generation of a species. Macroevolution is evolutionary change above that species level. So that's going to include things like mass extinctions, or so mass extinctions affecting the diversity of life and also the development of adaptations like flight, like back to our four-winged dinosaur example. So understanding macroevolution really has to get down to the business of geologic time. Now, I am not going to ask you to memorize the geologic time periods. The big things I want to show you in the next few slides is the fact that the continents that we know today have not always been that way. And one evidence for understanding how organisms evolved is to know that some continents which are very far apart today used to be very close together, or continents that used to be pretty far apart are now close together. So I do want you to see how continental drift affects things, but I don't want you memorizing any of the time periods. So I'm going to go through these, but again, don't focus on the time periods. I just want you to see how geologic change affects how organisms change. All right, so again, you look at the sequence in which fossils appear in rocks, and that gives you, sort of a family album of macroevolution. Now, you saw those layers of rock in the Grand Canyon. Well, here's the geologic time scale. Right, and so it looks like a giant blur. If you go way, 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 way down to the bottom, the stuff that's in tan, okay, it's showing the events that took place in terms of millions of years ago. Right, so 4,000 million years ago is going to be, 4 billion years ago. And if you look to that bar on the right, because to fit in words, they had to make the spaces relatively similar. If you look to the right-hand side, it shows the actual relative time span. So that Precambrian period, where we went from Earth forming to the oldest cells and the oldest animal fossils and all that. But look on that right-hand bar. That Precambrian period is most of the time span that we think of in terms of life on Earth. So it may be a relatively small bar on that left-hand side because they had to be able to fit text in there. But in real terms, if you look in the right-hand bar there, you can see how much of Earth's history was Precambrian and how relatively short the subsequent periods are. So again, you're not going to be memorizing Paleozoic, Mesozoic, Cenozoic, Anthropocene, any of that stuff. I mean, know the term Anthropocene, but I'm not going to have you memorizing time periods, to put it that way. But it's kind of neat to be able to see, the overall history. So how much time of the Earth was spent with just unicellular life, right? How long was prokaryotic things only? How long, when eukaryotic cells showed up, right? And then the oldest multicellular organism showing up. And so it's interesting to see how long some of these changes were. And then we get into the Paleozoic, and this is where I think I mentioned early in the semester the Cambrian explosion, the sudden divergence into all the living animal phyla that we're familiar with. Well, that's at the bottom of that. So you had that big event where there's a lot of divergence. And then from that divergence, all these interesting groups formed, right? So just sort of giving you the overall, situation here. All right, the Mesozoic era, that's the one most of us think of when we think of ancient life. This is what includes the Jurassic period. So this is where we have dinosaurs and gymnosperms being the dominant plants and the eventual formation of angiosperms. So this was a interesting period for people for a lot of reasons. But at the end of that, again, you had the major extinctions, right, that took out the dinosaurs, except for the birds and a few other species, and led us into the Cenozoic. All right, and this is where mammals really start taking over and angiosperm plants start taking over. And I'll talk about both these groups in the next couple of chapters. So just giving you sort of an quick overview of how things went.
By studying various fossil records, geologists have put together a geologic record. Evidence in the fossil record of large-scale extinctions and/or enormous radiation of many diverse new life forms defines the main boundaries within the geologic record. As you move through the geologic record, the names and dates will change. Also, a diagram at the upper left will show what is known of the history of continental drift, based on evidence dating from the Mesozoic era.
Top, you'll see what the
During the Precambrian time, which began at least 4,600 million years ago and ended 542 million years ago, life began on Earth. The first prokaryotic cells and cyanobacteria appeared, as well as the first animals. The beginning of the Paleozoic era, 542 million years ago, marked the origin of most modern animal phyla. The first plants, jawless fish, bony fish, and amphibians evolved. Amphibians became the dominant vertebrate life form, while the first reptiles and seed plants appeared. By the end of the era, 251 million years ago, many marine and terrestrial organisms had become extinct. The Mesozoic era stretched from 251 million to 65 million years ago. At the beginning of this era, the reptiles radiated out as dinosaurs to dominate the landscape, along with the cone-bearing plants, or gymnosperms. At the same time, the first birds and mammals originated. As the climate and Earth's surface changed, flowering plants appeared and many organisms, including most dinosaurs, became extinct.
The Cenozoic era followed the Mesozoic 65 million years ago and continues through modern times. The beginning of the era saw the radiation of the mammals, birds, and pollinating insects such as bees and butterflies. The angiosperms evolved to dominate the landscape as most modern mammalian orders appeared. The primate groups evolved, leading to the origin of ape-like human ancestors, and finally humans.
Okay, so you can see periods of time when you can review this, or if you go to johnkirk.com, there's really good animations of continental shifting. And again, it's not so much I want you to memorize what went where, but the fact that South America and Africa were nestled close to each other at one point, that it sort of explains some of the similarities that we see in groups. It wasn't that they moved from continent to continent. Those continents were once one continent as a single piece earlier. The fact that India was a free, like a giant island, and came up and crashed into the Asian continent, and is literally still crashing today. That's where the Himalayan mountains come from. So think of the most slow motion crash, where if you had two cars go together and you see the metal bending upward and upward and upward, that's what basically the Himalayas are. The mountain range formed when India did a slow crash into that continent. So anyway,
macroevolution by itself does not explain the diversity of life on Earth today. Let's investigate the role that macroevolution plays in this process. Multiplication of species creates biological diversity with the evolution of many different species from one common ancestor. This can be seen with Darwin's finches in the Galapagos Islands. Sometimes referred to as branching evolution or adaptive radiation, this occurs when multiple habitats and niches open up with relatively little competition. Evolutionary novelty results from the gradual refinement of existing structures for new functions. Here we see how ancestral birds may have used wing-like forelimbs for courtship. They could also have taken advantage of these enlarged forelimbs to allow for extended hops and gliding ability. Once flight itself became an advantage, natural selection would have remodeled feathers and wings to better fit their additional function. Explosive diversification often occurs following the evolution of an important adaptation. For instance, the evolution of the flower from early seed plants that were similar to modern conifers allowed plants to reproduce more efficiently and thus exploit many previously unavailable habitats and niches. After a mass extinction, previously filled niches and habitats are opened up. When the dinosaurs became extinct, mammals had the opportunity to evolve into those niches and produce incredible diversification.
Okay, so again, we're not going to get into memorizing all these areas, but I just want you to see sort of the large scale of evolution. So now one way we can learn how old something is by radiometric dating, and this is a way of observing how fast certain isotopes will decay over thousands, even millions of years. Another is to see layers of volcanic rock or ash above or below the layer where fossils are found. So in other words, if you can determine, okay, when this volcano occurred and maybe another volcanic event occurred, and you find stuff in the middle, then okay, the age of the fossil is between those two major geologic events. So the business of plate tectonics. So continents and seafloors are just a thin outer crust on this planet. And what happens is plates of that thin outer crust sort of float on top of the liquid mantle, right? So think of like a lava layer on the planet. And these big plates are drifting on that molten lava layer. And so the fact that movements in the mantle make those plates move, where plates are bounding, where plates come up against each other, there's usually a lot of geologic activity. So if you see something like earthquakes, two plates are scraping past or colliding with each other. Volcanoes form where you have edge of one plate shifting under another. So again, if you sort of look at Earth in cross section, that tiny gray layer is the crust, and so the plates of that crust are shifting around, sort of floating around on the mantle. And this diagram illustrates the tectonic plates, right? So if you look out at out to the right-hand side, right, there's that area of the Philippines up through Japan and down to north of Australia, right? Lots of volcanic activity. Well, that's where a series of plates are shifting and grinding against each other. You can see a similar thing out on the west coast of the United States, right? We talk about California and earthquakes. Well, if you look in the upper left, you can see where the North American plate is driving into, let's see, get the North American plate driving into the edge of the Caribbean plate. Any place where you see those little red dots, right, that's near where plates are interacting with each other. So why do you care about this in biology? Well, continental drift not only reshaped the planet, but it also altered the environment in which organisms live. So if you had something that lived, say, around the equator, and over millions of years, that location drifted upward, say northward, then what? Now the environment's going to change. The temperatures are changing. It alters the locations, because the animals are just along for the ride on these pieces of land. So the next couple of illustrations shows how plate movements formed this first huge continent, which reduced the total amount of shoreline, right? If you have a bunch of islands and you shove them all together, now you've less shoreline. It also allowed for ocean basins to become deeper and lowered sea level. So again, I don't want you to memorize these. I just want you to see this, because when I talk about some of the changes, it'll make more sense. If you look way down at the bottom, you see that one big island. And over time, that one big island splits into two other islands, right? Laurasia and Gondwana. And then those among themselves split. And so if you look, it's kind of hard to see, but if you look off where to the east of Africa, you can see India was an independent piece of land. And as we get towards the modern era, India drifted up and crashed into Eurasia and is literally still crashing today. It's a very, very slow crash. So again, I'm going to pop through these now because I don't want you to worry about this so much. This is where it makes any importance with biology. Understanding how continents moved around helps explain patterns of biogeography. And that's understanding the past and present distribution of organisms. Continental drift separated Australia from other land masses. And this is one of the reasons you see all the unusual marsupials and reptiles in some cases on Australia you don't see anywhere else in the world, because Australia was still attached to the major landmass when mammals first showed up. But before the mammals like us showed up, the eutherian mammals, the mammals that give live birth to fully formed young, well, Australia is separated before those eutherians showed up. So while marsupials were the dominant mammals, Australia separated. So when the rest of the world got taken over by eutherian mammals, these guys were safely tucked away further down in the Pacific Ocean. So that's one of the reasons why you see the unusual, mammal life there, because all these marsupials, which had cousins that were wiped out all over the rest of the world, are still there today. So if they originated in Asia and reached Australia via South America, if you look at how they're fitted together, the marsupials started in Asia and migrated west, crossed over South America and into Australia. So where would you expect to find fossil marsupials? South America, right? And so it also tells people, from knowing how the plates moved, gives you ideas of, hey, where should we look for transitional fossils for things? So again, here's some of our marsupials. And the mammals that we think of, most mammals that we know of are eutherians, right? Up around this part of the world, we tend to just see possums as the example of marsupials. But if you look on the left-hand side, you've got a quoll, which is a small carnivore. So think of the way that we have small cats or weasels, right? We have small mammals that hunt other animals, hunt insects, things like that. Look on the upper right rather. You have a sugar glider, which looks a lot like a flying squirrel. Well, in some respects, they share some anatomy, but not because they're close relatives. But they lived in similar enough environment that they appeared, to take similar form. I'll get into that in a little bit too. Now, mass extinctions, again, we're not going to be memorizing these, but again, most everyone knows about the Cretaceous extinction because they lost all the dinosaurs except the birds, right? And that's when mammals took over the place. But there's been 5 major extinctions. Some people consider the modern era the 6th extinction, but we won't get into that too much here. But just to show you like how mammals evolved after the dinosaurs were wiped out. So you see the reptilian ancestor. Mammals evolved from reptile ancestors, right? And you have those first mammals. And mammals are kind of oppressed by dinosaurs, right? Dinosaurs were the dominant vertebrate forms on land. So most mammals were small, ran around at night, tried to keep away from the dinosaurs and turning into dinosaur chow. So if you follow the line of these ancestral mammals, they're very narrow, right? There's not a lot of forms until you get to about 66 million years ago. And that's when the dinosaurs got wiped out. Now look at how the bottom 2 lines widen, right, into much broader lines. That's because of the massive diversification that took place in marsupials and eutherians. The monotremes up at the top were a small group and remain small today. In chapter 16, I'll get into, no, chapter 17 rather, I'll get into all these guys. So one thing we talk about was, all right, biogeography is a factor we consider when studying macroevolution. Another is, if you think about the different forms of things, more and more scientists can explain things that happened at the macroevolutionary level in terms of genetics, right? You could see changes in the fossil record. You could see different things in the fossil record, but how did you get from one form to the other? So there's now this area called Evo-Devo. it's basically where evolutionary biology meets with developmental biology, right? What happens between a gamete, between a zygote and the fully developed organism. And slight changes in how genetic information flows can get magnified into big structural differences. So evolutionary transformations, changes in organisms, can result from genes that alter the rate, the timing, and the spatial patterns that take place as an organism transitions from a zygote to an adult. Great example is looking at humans and chimpanzees. Genetically, we are very, very, very similar. So why is it we look so different? Well, if you look at skull development, in the left-hand side you see two fetuses, the upper one chimpanzee, the lower one human. And they look pretty similar, right? Well, all those little boxes sort of map out those areas of those two skulls. What happens during development shows what happens to those squares. So some are going to become much, much larger and stretch out. And so the changes in development from a very similar fetal form to what you see on the right-hand side, where if you look at the human skull, the cranium, the dome of the top of the skull, balloons outward, whereas it doesn't do so in the chimpanzee. The chimpanzee's lower jaw, where it becomes very pronounced and the teeth become much more heavily developed than you see in the human, right? So it's kind of a neat theory that humans actually more closely resemble the fetal skull, right? If you look at the human skull versus the fetal skull, adult versus fetal, and then the chimpanzee adult versus fetal, it's thought that humans basically changed to basically retain more baby-like features. The high forehead, the domed skull, the small jaws. Now, I won't drive on with this, but it's actually a fascinating field. And grad school actually had an entire seminar just on Evo Devo, so I'm going to step away and let you guys go. But hopping over here, so biological novelty. Adaptation of old structures for new functions. And the feathered, nature of birds and the fact that birds fly is the way that we know is a great combination of structure and function. Because in flight, the shapes and arrangements of various feathers on a bird's body produce lift, produce smooth flow of air over the wings, and even help with steering and balance. So where the heck did this come from? Well, as you know from the four-winged dinosaur video, which hopefully you all have had a chance to see already, thousands of fossils of feathered dinosaurs have been found. Right, we're finding more and more of them, a lot of them coming out of Asia as there's been more fossil investigation in Asia. And they found these fossils and been able to classify them into 30 different species. For the longest time, there were only a few fossils out of Europe, but now we have many more to work from. But the feathers you see in those fossils couldn't have been used for flight. They weren't flight surface type feathers. And if you look at the limbs of the animals, they weren't quite suited for flying. Right, and this is Archaeopteryx. This is a great example of an ancient bird, which you hopefully saw in the video. Now again, that long bony tail, that's a very reptile thing. Birds don't have long bony tails. Teeth are things that you see in reptiles and not in typical birds. But you see those elongated forelimbs, right? So it's reptilian, but it's the fact that you're getting that fusion of the wrist and those elongated fingers tells you something different is going on. And you can see the impressions of the feathers all around there. So what were feathers first for? Possibly insulation, right? Keeping warm. Once flight itself became an advantage, then natural selection would have gradually remodeled feathers and wings to fit their additional function. So again, natural selection's not guided. It's just mutations that improved feathers or improved limbs to form wings. Those mutations were selected for. So again, it wasn't an intentional thing. It's just mutations that favored these things were favored in terms of those organisms reproducing and changing the allele frequency in those features over generation to generation. Now, there's a term, structures like feathers that evolve in one context but become co-opted, used in another function, are called exaptations. So something where a feather might have had one function like keeping warm eventually became a flight surface. So that would be a great example of exaptations. All right. So from simple to complex structures in gradual stages, there's conflict in people who study evolution. Some people think complex structures evolved in little bitty steps, and there are examples of that. And some people feel like giant changes, like think of a mutation that radically changed the limb bones in what would become wings in birds. So something where there was a radical change that led to a radical difference, and you saw a change happening that way. Most of the time, we see complex structures evolving in small steps, right? So from a simpler version with a basic function to slightly more refined functions, slightly more complex functions developing. So usually we think in terms of gradual evolution. And the evolution of complex eyes is a great example of this. You can see evolution from a simple little patch of cells that reacted to light, right? So basically telling light from dark. Over time, mutations that led them to be more sensitive, to be able to receive wider range of wavelengths, to be able to determine images. These are all changes that benefited their owners. Now, there are some organisms that never develop fancy eyes, right? So let me go to this next picture here. So you have a mollusk on the left called a limpet, another mollusk called an abalone on the next to the left on the left side. The one in the middle is a nautilus. And cephalopods, as you hopefully know from the video, have very complex eyes. And then you see other eyes in marine snails, and then onward to the really great eyes of squid, right? So all of these are mollusks. And you see the most simple eyes in the limpets, where basically just a little layer of cells that can receive light and take some information from that. But as you keep moving from left to right, you can see that the structures become more complicated, and with it, when you start seeing things like the development of things like a lens, like a protective surface over the lens, you start seeing these more complicated structures. And if you remember looking at things like cuttlefish, those cuttlefish, and that includes the same group as squid, have very complex eyes, right? The fact that they could see a surface and be able to change their shape to mimic that surface, right? That takes good visual function. So just a nice example of showing you gradual change, and this is within just one group of animals, right? Just only within the mollusks. So again, you're not memorizing all the anatomy here. I just want you to see a nice example of gradual changes leading to more complex eyes. All right, so taxonomy. So we mentioned this a little bit in the last chapter. It comes back again here. Taxonomy is again, the naming and classification of species. Systematics includes taxonomy and focuses on classifying organisms and figuring out what the evolutionary relationships are. So systematics involves naming, but it also involves how do you separate them into groups to give names, right? So biologists use phylogenetic trees to show hypotheses about the evolutionary history of species. Because remember, there's no answer book to any of this stuff. So every time you develop a pattern, you say, okay, this is the relationship of this organism to that organism, and this was the ancestor to this, and how these things are related. These are all hypotheses. These are all an answer to the question, how are these organisms related? So people spend large chunks of research figuring out what features are best to use to categorize things, what features are more important than others, how do you develop these phylogenetic trees. So it's a lot of work. We only see the finished products, but to get there is often years of research. Now that we have computers, we can calculate possible trees a lot faster, but it still takes quite a bit of time. Ideally, these hypotheses, these hypothetical phylogenetic trees, show the evolutionary history of species, right, from ancestor to descendant. and also reflect that classification of groups. Remember that whole King Philip came over for good spaghetti, right? How do you determine what classes are and what classes fit into, a particular family of organisms, right? So reflecting the groups that contain other groups. Now, back to our carnivores here. So you had order Carnivora, which is divided into three families: Felidae, Mustelidae, and Canidae. Genera, right? We're just looking at a few now. There are many more genera within these families. They're just showing you a few just to give examples. But Panthera within the Felidae, Mephitis and Lutra within the Mustelidae, and then Canis within the Canidae. And then you've got those species, and the word species is spelled wrong on your slide. Okay, I have to fix that. You've got Panthera pardus, Mephitis. You've got the leopard, the striped skunk, the European otter, the coyote, and the wolf, right? So you can see just on a large scale that, oh yeah, there's three major physical forms here: the cats, the weasels, and then the dog-like animals. So further than this, you have other types of skunks, other types of otters, and people have to sort out, how are they related, which groups contain smaller groups, and things like that. So why do we care about phylogeny? Well, there are practical uses to this. They give an example here. They figured out the phylogeny of maize, right, the ancestry of domestic corn. And they've identified two species of wild grasses that were probably the closest relatives that are still around today. And how do you do this? Well, you compare the genomes. And so the genomes of these plants may hold alleles that offer disease resistance or other useful traits that can be transferred into the maize that we have cultivated today, the corn we have cultivated today. And why is that interesting? Well, other than the sheer science, look around us. The world's changing. If we can find alleles that can help us adapt our cultivated plants to the changing world. So, say one group of people is studying rice, and rice is typically grown in very wet, very warm areas. Well, they're trying to find alleles to allow rice to grow in drier areas and possibly cooler areas. So as climate change shifts further and further, maybe we're going to see a shift from wheat to rice as things get warmer and warmer. So it's kind of neat that people not only to understand how these things form, but say, okay, let's find alleles that we might want to use later. It's another good reason to conserve the ancestral forms of a lot of our domesticated organisms, because you kind of want to have the raw material and maybe have to call back to some of those earlier alleles. Okay, moving on from there. Homologous characters. This is a popular one with the people sorting out systematics. Homologous, so homo meaning same. Homologous structures in different species may vary in form and function, but have similarities because they evolved from the same structure in a common ancestor. So again, think back to the forelimbs of vertebrates that we had. We had the human, the cat, the bat, and the whale, right? So those are four mammals. And you could see the homologies in those forelimbs. They evolved into different functional structures, but you can still see the humerus, you can still see the radius and ulna, etc. And homologous structures are great for sorting out phylogenetic relationships. But there's another thing that takes place, and this is called convergent evolution. Right? Convergent means things coming together to a single point. So convergent evolution involves superficially similar structures from different evolutionary branches that result from natural selection shaping analogous adaptations. So similarity that's due to convergence is called analogy, not homology. So the wing of a bird and the wing of a bat, right? Those two structures evolved in two different tracks of organisms. But to fly, there are certain features that tend to be in common, right? To have something where you have an elongated limb and a flight surface. Now, the flight surface in birds are feathers. The flight surface of a bat is skin, right? But at first you'd say, oh, these two things both have wings. So you can have these things that look similar superficially, but the organisms bearing them are not closely related. That little sugar glider that you saw back on the back when I was talking about the Australian mammals, there are other things that have similar lives and have evolved similar anatomy, even though they're not closely related. That sugar glider has a lot in common with a flying squirrel, which is something we have here in North America. They're not closely related at all, right? You have a marsupial and a placental mammal, but because they have some similarities in how they live, you see some similarities in their anatomy. So comparing the comparing the embryonic development of two species can often reveal homologies that aren't apparent in the mature structures, right? Think about comparing things in the form of the fetus, right? So if homology reflects common ancestry, then comparing DNA sequences, right? Now you're drilling down even further, right? You're not looking at structures that might have evolved in different environments or whatever. You're comparing the actual genes, right? So if you can compare DNA sequences of organisms, then you're really going to get to what they call granular, fine detail understanding of evolutionary relationships. So the more recently two species branch from each other, the more similar their DNA sequences are, like humans and chimpanzees. And sometimes we have fossils that are preserved so well, like ancient birds being fossils of ancient birds being found in these lava tunnels under New Zealand. Well, some of those birds were never disturbed once they had died down there, and they could actually extract DNA fragments that could be used to compare to living organisms today. Now, cladistics. This is a way of grouping organisms in classification. And this is, again, grouping things by common ancestry. A group is going to be a common ancestor and everything that descended from it. So a clade consists of an ancestral species and all its evolutionary descendants, and it forms a distinct separate branch in the tree of life. So when you can identify clades, when you can identify ancestors and all their descendants, then it helps you construct good classification schemes, right? It actually shows the pattern of how things went from ancestral forms diverging into other species, other forms. And again, cladistics comes back to that Darwinian concept, right? Descent with modification from a common ancestor. Now, to identify clades, it gets into things like points of reference, right? You compare an in-group with an out-group. The in-group is the group of species you actually want to analyze. The out-group is a species or group of species that diverged before the lineage you're studying, right? So if a lineage is an ancestor and all the descendants, this is one that's similar to the original ancestor. And let me show you in the next diagram. So this is a real simple example. You see that that initial little branch on the initial little stem on the left, right? That group of organisms gave rise to both reptiles and mammals. Now, what we want to study are the mammals. What we do is we take a close relative to their ancestors to compare, right? So we can see things that are going to link together all these mammals, the in-group. But one thing you can compare them against are animals like reptiles like iguanas, right? So you use an outgroup to compare. So having 4 legs is something all of them have, right? So you wouldn't separate any of those mammals on the basis of having 4 legs, because even their ancestors and the other descendants of their ancestors had 4 legs, right? So 4 legs would not be a distinguishing characteristic. Hair, that would be a distinguishing characteristic, right? That's something that those mammals all share that are not shared by the outgroup, the reptiles, right? Gestating the young inside the body versus eggs would be something that separate the kangaroos and beavers from the duck-billed platypus, which actually is an egg-laying mammal. And also the fact that laying eggs is something that you'd say, wait, iguanas and platypuses both lay eggs. Maybe they're in the same group. But you also have the hair and mammary glands separating them. So there are some things that may seem to be causing different linkages, but you separate them by a whole pile of other reasons. So you separate the iguanas from the mammals by hair and mammary glands. Within that group of things that have hair and mammary glands, you separate one group that lays eggs from the groups that maintain, that gestate the offspring inside their body. Okay, out of things that gestate their offspring inside their body, you separate two groups. One that basically give birth to not quite done yet offspring versus ones that hold the offspring in long enough and it develops into a more complete individual. So there's things you can do to hold together the in group, and then there's the features that separate members of the in group. So it's kind of neat in that this clarifies stuff that, in ways that weren't apparent in other classification systems. And the best one that most of us can observe right around us is birds. Originally, birds and reptiles were placed in separate classes of vertebrates, right? So classes are these subgroups of vertebrates. Sorry, start that back. Biologists traditionally place birds separately. Now, if we pile up the homologies looking at birds, mammals, reptiles, we realize that birds and crocodiles actually are in the same clade. They share all these features that separates them from the other reptiles. So another clade would contain the lizards and snakes. Now, if you go far back enough, as the ancestor that crocodiles share with lizards and snakes make up the clade, right? So the ancestor that gave rise to crocodiles, dinosaurs, and birds, and lizards and snakes, right? If all these guys share one common ancestor, then class Reptilia has to include the birds. So you have that furthest most stem on the left-hand side, right? That was the ancestor of all reptiles. Now, reptiles diverged into two major groups, the lizards and snakes, and all these other guys. So at that next branch point, you see the common ancestor of crocodilians, dinosaurs, and birds. So if you have that ancestor and count all of its descendants, you have to include birds, because you couldn't just pull birds out and give it its own little branch. No, that ancestor gave rise to all those four groups on the right-hand side. So we now know that we have to include birds when we talk about dinosaurs. So sometimes you'll hear me say non-avian reptiles, right? So reptiles that don't include birds. Now again, phylogenetic trees are hypotheses, right? These are people trying to sort out evolutionary history. And again, the more information we get, the more we can fine-tune these hypotheses. Now, Carl Linn, otherwise known as Carolus Linnaeus, he divided all known forms of life between plant and animal kingdoms, because that's what people understood life to sort into. 20 questions, animal, vegetable, or mineral, right? The living things only came in animal or plant flavors. That two-kingdom system prevailed for over 2 centuries. In the mid-1900s, that two-kingdom system was replaced by a 5-kingdom system that put all the prokaryotes in one kingdom and divided the eukaryotes among 4 other kingdoms. So we now have 5 kingdoms. Now we got to the late 20th century, molecular studies in cladistics led to the development of a 3-domain system that consisted of 2 domains of prokaryotes and 1 domain of eukaryotes. Now, the domain eukarya is divided into kingdoms, but you could argue the exact number of kingdoms, right? So we have here the 3-domain system. So earliest organisms, the first branch gave rise to the group that became bacteria and the group that became the ancestor to archaeans and eukaryotes. So let's go down the eukaryon line. And you see here kingdom Animalia, kingdom Fungi, kingdom Plantae, and the protists. Because some books will say kingdom Protista, but you really don't have a tidy classification. It will probably, in generations to come, that's probably going to get divided into more kingdoms. So luckily for you, you just have to know them as one big garbage can of protists. But as time goes on, they're going to have to find a way to sort out the relationships within that crowd, and it's probably going to lead to multiple kingdoms within that. Okay, so last couple of slides. So back to the Anthropocene. So this is a time of huge environmental change, right? Habitats are being affected by us. And as we've changed the world around us, we're actually being able to observe evolutionary adaptation because the changes are so rapid because of what we've done. It's not like slow geologic changes. We're causing such radical change that we're seeing evolution taking place within all these habitats around us. So things like resistance to toxic pollutants, crested anoles in Puerto Rico, these are these little lizards, they have more grippy scales. Why? Well, because the ones that live in cities are not climbing up rough plants and trees. They're climbing up thin, smooth, slippery surfaces like concrete and metal. So you compare those guys that have generation after generation in a city versus the ones that never moved, never lived in the city. You actually see physical changes in the scales on the bottom of their feet. Urban blackbirds sing at a higher pitch. Why? Because in the city you have traffic noise, and a low pitch call would be lost in all the traffic noise. So people who study the bird calls, the ones that live in cities sing at a higher pitch so they can hear each other over traffic noise. So urban adapted animals are a whole area of study and seeing how animals are adapting to this new environment. So one group of birds and lizards get blown out to an island, another live in the city. And that's the end of the chapter.